What is conscious evolution? I'm your host, Bruce Lipton, for this series on inner evolution. The fractal character of nature offers deep insight into understanding how to live in health and harmony by studying the behaviors of other forms of life. Human hubris leads us to believe that we are the highest level of earthly intelligence, and as one descends through the lineage of lower animals going toward the base of the tree of life, each is endowed with lesser and lesser intelligence. Single cells are near the base of this tree. How much intelligence could a cell have? Conventional perception would say very little, if any. Guess what? The community of single cells were smart enough to make humans using technologies that we cannot yet fathom. In this session, we use nature's fractal geometry to gain insight on the mechanics of evolution. The metric of evolution is not based on genes, but on the development of the nervous system. Starting with the primitive prokaryote organisms, we can assess the hierarchy of evolution by measuring an organism's membrane surface area. This approach not only illuminates the pattern of past evolutionary advances, more importantly, we can use the same assessment to project the exciting vision of our future, conscious evolution. In previous episodes, what we really discussed was the nature of how cells work, where intelligence comes from. At first, it was thought that intelligence was based on the number of genes an organism has. And this is actually what led scientists to pursue what is called the Human Genome Project. In the failure of finding 100,000 anticipated genes in the human, it was necessary to go back and consider what is evolution actually based on. And in 1809, a scientist in France, Jean-Baptiste de Lamarck, was the first scientist to actually write a theory of evolution. And in his theory, Lamarck said evolution was reflected in the development of the nervous system. Well, we can look at that same understanding today and say, well, yes, humans have indeed a profoundly more effective nervous system than lower organisms. But there's a problem. How do you measure intelligence in a nervous system? Well, as we discussed earlier in this series, all cells have intelligence. So we don't have to look at the human. We could go down and look at a very primitive cell and say, what is intelligence in a primitive organism? In our effort to understand that, we started to focus on the cell membrane, the skin of the cell. And we found that there are proteins built into the membrane which consist of a receptor and an effector protein. And we looked at these protein complexes of the receptor effector, and guess what we recognized? By definition, the receptor represents awareness of the environment through physical sensation. The significance of this definition is it is the exact same definition to describe the word perception. So instead of calling these proteins a receptor effector integral membrane protein complex, we can simply refer to these proteins as perceptions. Significance of that is a perception is a foundational unit of consciousness. So as perceptions increase, so is there an increase in consciousness. Significance is profound because basically it says if you want to measure consciousness, then the fundamental unit of consciousness is a unit called a perception unit, which is a receptor effector protein complex. Now it becomes interesting because if we want to measure consciousness in evolution, what we're really going to measure is the increase in these protein complexes. In our previous discussion, we also recognized the fact that the membrane, by definition, is an information processor, the equivalent of a chip, and that the receptor effector proteins are the equivalent of a bit of data. And when we talk about computers, we can talk about how intelligent is your computer, and the answer comes down to how many bits of data can your computer handle? Well, it actually applies exactly the same to organisms. In other words, simply this: how many bits of data in a membrane does a cell have? So all of a sudden we say, now there's a physical metric to measure consciousness, the receptor effector units. Well, the significant aspect of reading how many of these units are found in a membrane is a very interesting concept based on how can these proteins fit into the membrane. Significance is this: the membrane has a thickness of about 10 nanometers, and this is a dimension defined by the lipid molecules that create the basic structure of this membrane barrier. Relevance is the protein's bits that we're talking about have essentially the same dimension. This becomes very important because it says this: if you want to add more bits of proteins, more perception into the membrane, you can't stack the proteins on top of each other because the size of the membrane is previously defined by the phospholipids. You can only lay them out in a flat surface of the membrane. I go, wow, well then you understand this fact. The number of bits in a membrane is determined by the surface area of the membrane. The more membrane there is, the more perception bits I can put into the membrane. I can measure consciousness by measuring an organism's surface area. But now, when we go back and look at the evolution, we start off with a very primitive organism, and that is the prokaryote, the bacteria. And I say significance of this is that this is the first organism, and it also has consciousness. Yes, a bacterium can be aware of the environment and make an appropriate life-sustaining response, which is the definition of consciousness, awareness of the environment. And the awareness of the environment, of course, is due to the protein bits built into the cell membrane. So we want to measure the surface area of a bacterium membrane, and that will give us a readout of how much consciousness the bacterium can have. Well, bacteria are like invertebrates, like insects. They have a hard outer shell. It's actually called a capsule. And the capsule defines the size of a bacterium. Significance? The size of a bacterial cell is limited by this external capsule. If you look underneath the capsule, you'll see the membrane of the bacterium, and you can calculate the surface area and then get an idea how much consciousness this bacterium expresses. And as bacteria evolved, they actually expanded their membrane a bit, causing the membrane to fold inside of the cell, as illustrated in this picture. This folding of the membrane is called a mesosome. Significance? It expands surface area. Relevance? More surface area, more intelligence. However, there's a problem. You can only put so much membrane within the capsule of the bacterium. And it basically said this: when bacteria were created, the membrane representing consciousness was increasing as the bacterial cell gained more consciousness, but then it reaches a finite size. You can't put any more membrane in a bacterium. Well, wait a minute. If the expansion of the membrane is an expression of evolution, and all of a sudden a bacterium reaches a finite size and can't put any more membrane in it, my goodness, what this really represents is that evolution stopped. It couldn't make a smarter bacterium. But obviously evolution didn't stop. What happened is there a second phase of evolution. In phase 2 of evolution, bacteria came together in community to share awareness. And this made a more intelligent community. And then we recognize in our evolutionary story, bacteria communities surrounded themselves with a membrane. And in this membrane were not just the same bacteria, they were a mixture of bacteria with different bacteria expressing different functions, but by collaborating, the community was more advanced than any single bacterium could have become. So evolution now has 2 phases. Phase 1, make the smartest individual cell, and in phase 2, bring the smartest individuals together and create a community, because in community, shared awareness could advance the intelligence of the group. So, what do we have? First level of evolution, single bacteria. Then we made the smartest bacterium. They formed a community, surrounded themselves with a membrane, and in biology we call this a biofilm, a heterogeneous community of bacteria living in harmony. Well, is that the evolution end? No. What happened is nature made the system more efficient. Rather than each bacterium living on its own in the community and providing some function, what happened over evolution was this. The genes of the individual bacteria were coalesced into a single structure called the nucleus. And so now all of the genetic activity of all the different bacteria are coalesced into one nucleus containing all of the information. And then all of a sudden, this former community of biofilm with a membrane around it turns into a new organism. It becomes what we refer to as a eukaryote, an organism that has a nucleus in it. The nucleus is the repository of all the genes of the former community. In addition, something new happened. Instead of putting a capsule to contain this community, actually an internal skeleton was created. Because with an internal skeleton, you can expand the surface area, where you couldn't do that when there was a hard shell outside of the cell. So with an internal cytoskeleton, the new cell that formed, the eukaryotic cell, is represented by something that we would call an amoeba. An amoeba is a new identification. An amoeba represents an advanced evolution of a former community of bacteria. But now it's a single organism. And I go, so what? Well, with the expansion of a cytoskeleton, you can use the membrane like a tent. The more cytoskeleton, the more surface area the cell can have. So a single amoeba has 1,000 times more surface area than a single bacterium. Since surface area is a measure of consciousness, then all of a sudden you can recognize the amoeba cell is 1,000 times more conscious than a single bacterium. So what happens in the next phase of evolution? You make the smartest amoeba possible by enhancing the membranes of this structure.